As you settle into this quiet moment, perhaps you notice the subtle sensations within your body, the gentle fullness or emptiness of your abdomen, the faint warmth radiating from your core. Right now, without any conscious effort from you, a remarkable system is at work, breaking down nutrients, absorbing what sustains you, eliminating what you no longer need. This process has been continuous since you last ate, and it will continue through the night, through tomorrow, through all your days. The gastrointestinal system, that long and winding passage from mouth to exit, performs one of life's most fundamental transformations, turning the external world into you. Tonight, we explore this elegant digestive architecture, a system that spans nearly 9 meters from beginning to end, folding and coiling within the compact space of your abdomen. This is not merely a tube for processing food, but rather a sophisticated chemical factory, an immune sentinel, a microbial ecosystem, and a sensory organ all woven together. Every meal you consume begins a journey through environments as varied as any landscape, from the mechanical grinding of teeth to the acid bath of the stomach, from the enzymatic rivers of the small intestine to the slower, drier passages of the colon. We will move through this exploration gently, discovering how the simple act of eating initiates cascades of hormones and enzymes, how the body distinguishes nourishment from threat, how trillions of microorganisms participate in your digestion, how nutrients cross from the external channel of the gut into the internal rivers of your blood. This journey requires no effort from you, no memorization, no testing of knowledge. We are here simply to appreciate the quiet intelligence of a system that has evolved over hundreds of millions of years to extract life from the world around us. Think of the gastrointestinal tract as a great river flowing through the landscape of your body. Like a river, it has its source, the mouth where water and solids enter. It has rapids and falls, the muscular contractions that propel contents forward. It has deep pools where matter settles and transforms, the stomach holding food for hours. It has broad deltas where tributaries merge, the ducts carrying bile and pancreatic secretions into the intestine. And it has a mouth where it meets the sea, the final exit where the unusable remnants return to the external world. Throughout our exploration, this river metaphor will help us understand the flow, the chemistry, the ecology of digestion. You may notice, even now, the gentle movements of your digestive system. Perhaps a subtle gurgling, a sense of shifting deep within your abdomen. These are the sounds and sensations of peristalsis, the rhythmic muscular waves that push contents forward, that mix and churn and gradually propel what you have eaten through its transformative journey. This movement never truly ceases, whether you are aware of it or not, whether you have recently eaten or your stomach is empty. The river flows on. Let us begin gently with a question that opens into depth. What exactly is the gastrointestinal system, and what purposes does it serve? The answer unfolds in layers, each revealing new dimensions of function and complexity. At its most fundamental level, the gastrointestinal system is a hollow tube running through the body, technically remaining outside the body's true internal environment, even though it exists within the physical boundaries of the organism. This tube, also called the alimentary canal or digestive tract, begins at the lips and ends at the anus. Along this passage, the external world, food and drink, encounters a series of specialized environments designed to break it down into absorbable components. The primary function is digestion. the mechanical and chemical breakdown of complex food molecules into simpler forms that can be absorbed. Proteins become amino acids. Carbohydrates become simple sugars. Fats become fatty acids and glycerol. This breakdown is essential because only small molecules can cross the intestinal lining and enter the bloodstream. The steak you ate for dinner, the bread, the vegetables, all these complex structures must be dismantled into their molecular building blocks before your body can use them. Absorption represents the second major function, the transfer of these digested nutrients from the gut lumen across the intestinal lining into the blood or lymph. The small intestine particularly excels at absorption, its lining specialized with microscopic projections that vastly increase surface area. Through these surfaces, sugars and amino acids enter capillaries that carry them to the liver. Fats enter lymphatic vessels that eventually drain into the bloodstream. Water, vitamins, minerals, all must cross this critical barrier. Motility describes the movements that propel and mix contents. Chewing breaks food into smaller pieces. Swallowing pushes the bolus from mouth to stomach. Peristaltic waves, coordinated muscular contractions, drive contents through stomach and intestines. Segmentation contractions mix contents without moving them forward. These varied movements ensure that food contacts digestive enzymes, that nutrients encounter absorptive surfaces, that waste materials eventually reach the exit. Secretion encompasses the release of enzymes, acid, mucus, bile, and other substances into the digestive tract. Salivary glands produce saliva containing digestive enzymes. The stomach secretes hydrochloric acid and pepsin. The pancreas contributes a rich mixture of enzymes. The liver produces bile for fat digestion. Specialized cells throughout the tract secrete mucus that protects the lining and lubricates passage. These secretions create the chemical environments necessary for digestion. Beyond these classical functions, the gastrointestinal system serves as a major immune organ. More immune tissue resides in and around the gut than anywhere else in the body. The intestinal lining must distinguish between nutrients that can be absorbed, harmless microorganisms that form the normal gut flora, and pathogens or toxins that threaten health. This immune surveillance operates continuously, sampling the gut contents, mounting responses to threats while tolerating the beneficial and the benign. The gastrointestinal tract also functions as an endocrine organ, producing numerous hormones that regulate digestion, signal satiety or hunger, influence metabolism, and even affect mood and cognition. Cells scattered throughout the intestinal lining sense nutrients and release hormones that coordinate digestive activities. The gut produces more serotonin than the brain, contributing to the bidirectional communication between gut and central nervous system, sometimes called the gut-brain axis. The ecosystem of microorganisms inhabiting the gut, collectively called the gut microbiota or microbiome, represents another dimension of gastrointestinal function. Trillions of bacteria, along with archaea, fungi, and viruses, live within the intestinal tract, particularly the colon. These microbes help digest certain foods, produce vitamins, protect against pathogens, and influence immune development and function. The relationship between human host and microbial inhabitants is deeply mutualistic, with each supporting the other's existence. Understanding the gastrointestinal system therefore requires appreciating this multidimensional nature. It is mechanical and chemical, absorptive and secretory, immune and endocrine, human and microbial. It exists at the interface between self and other, between internal and external, continuously making decisions about what to accept into the body and what to reject or eliminate. The importance of this system becomes clear when we consider that every molecule of glucose fueling your brain, every amino acid building your proteins, every fatty acid forming your cell membranes, every vitamin enabling enzymatic reactions, all these entered your body through the gastrointestinal tract. You are literally made from what you have eaten, transformed through digestion and absorption into the substance of your cells and tissues. The old saying proves true: you are what you eat, or more precisely, you are what you digest and absorb. Now let us examine the basic architecture of this system, the anatomical structures that form the digestive tube and its associated organs. The gastrointestinal tract proper consists of the continuous hollow tube with specialized regions, while accessory organs, the salivary glands, liver, gallbladder, and pancreas, produce secretions that enter this tube and aid digestion. The wall of the digestive tube, from esophagus through intestines, follows a consistent organizational pattern with 4 main layers. Understanding this wall structure provides insight into how the tube performs its various functions. The innermost layer, facing the hollow space or lumen, is the mucosa. This layer itself has 3 components. The epithelium, a layer of cells forming a continuous lining, directly contacts whatever passes through: food, digestive secretions, microbes. The epithelium varies along the tract's length according to function. In the mouth and esophagus, it is stratified squamous epithelium, multiple layers of flat cells resistant to abrasion from food. In the stomach and intestines, it becomes simple columnar epithelium, a single layer of tall cells specialized for secretion and absorption. Beneath the epithelium lies the lamina propria, loose connective tissue containing blood vessels, lymphatic vessels, and immune cells. This layer supports the epithelium and houses much of the gut-associated immune tissue. Below that is the muscularis mucosae, a thin layer of smooth muscle that creates small folds and movements of the mucosa, helping mix secretions with contents and maintaining contact between epithelium and luminal contents. The second major layer is the submucosa, dense connective tissue containing larger blood vessels and lymphatic vessels, nerve networks called the submucosal plexus, and sometimes glands. This layer provides structural support and houses the vascular and neural infrastructure serving the mucosa. The third layer is the muscularis externa, typically consisting of 2 layers of smooth muscle. The inner layer has fibers arranged circularly around the tube. The outer layer has fibers running longitudinally along the tube's length. Between these muscle layers lies another nerve network, the myenteric plexus. The coordinated contraction of these muscle layers produces the movements of the digestive tract. The outermost layer varies depending on location. In the abdominal cavity, it is usually serosa, a thin layer of connective tissue covered by a smooth membrane, continuous with the peritoneum lining the abdominal cavity. In areas outside the abdominal cavity, like parts of the esophagus, the outer layer is adventitia, connective tissue that blends with surrounding structures. This 4-layer organization, mucosa, submucosa, muscularis externa, and serosa or adventitia, creates a tube capable of multiple functions. The epithelium provides a selective barrier and performs absorption and secretion. The muscle layers generate movement. The nerve networks coordinate activity. The blood vessels supply nutrients and remove absorbed substances. This design repeats with variations throughout the digestive tract, a modular architecture adapted to local needs. The enteric nervous system, composed of the nerve networks in the gut wall, deserves special mention. This is sometimes called the second brain because it contains as many neurons as the spinal cord and can control digestive functions independently of the central nervous system. The enteric nervous system coordinates muscular contractions, regulates secretions, senses chemical and mechanical stimuli in the lumen, and communicates with the brain through the vagus nerve and other pathways. This intrinsic nervous system allows the gut to respond locally and rapidly to changing conditions while also integrating with central control. Now we trace the passage from mouth to exit, examining each region's specialized structure and function. The journey begins with the oral cavity, bounded by lips, cheeks, palate, and tongue. Here, mechanical digestion commences with chewing, also called mastication. The teeth, anchored in the jawbones, cut and grind food into smaller particles. Humans have different tooth types suited for different tasks: incisors for cutting, canines for tearing, premolars and molars for grinding. The tongue, a muscular organ covered with specialized epithelium, manipulates food during chewing, mixing it with saliva and positioning it between teeth. The tongue also contains taste buds, sensory structures that detect sweet, sour, salty, bitter, and umami tastes. Taste provides pleasure and helps assess food safety, with bitter tastes often warning of toxins and sweet tastes indicating energy-rich nutrients. Saliva, produced by 3 pairs of major salivary glands and numerous minor glands, begins chemical digestion and facilitates swallowing. The parotid glands located near the ears produce watery saliva rich in amylase, an enzyme that begins breaking down starches. The submandibular and sublingual glands, located beneath the jaw and tongue respectively, produce saliva containing both enzymes and mucus. Together, these glands produce about 1 to 1.5 liters of saliva daily. Saliva serves multiple functions. It moistens food, making it easier to chew and swallow. It contains amylase, initiating starch digestion. It dissolves food chemicals so they can reach taste buds. It contains antimicrobial agents that help control oral bacteria. It buffers acids, protecting tooth enamel. It lubricates the oral cavity and esophagus. Saliva production increases in response to food in the mouth and even in response to the sight or smell of food, demonstrating the anticipatory nature of digestive regulation. When chewing is complete, the tongue shapes the chewed food and saliva into a cohesive mass called a bolus and pushes it backward toward the pharynx. This initiates swallowing, a complex coordinated sequence involving voluntary and involuntary components. The voluntary phase propels the bolus from mouth to pharynx. The involuntary pharyngeal phase then takes over, a rapid sequence lasting about 1 second where the soft palate rises to seal off the nasal passages. The larynx elevates and the epiglottis folds down to protect the airway, and muscular contractions propel the bolus into the esophagus. Breathing briefly pauses during swallowing, coordinating these 2 functions that share the pharynx. The esophagus is a muscular tube about 25 centimeters long, running from the pharynx through the neck and chest to the stomach. Its wall follows the basic 4-layer pattern, with stratified squamous epithelium in the mucosa resistant to abrasion, and with both skeletal and smooth muscle in the muscularis externa. The upper third contains skeletal muscle under voluntary control. The middle third transitions between muscle types, and the lower third is smooth muscle under involuntary control. Peristalsis, the primary movement pattern of the esophagus, consists of waves of contraction that push the bolus downward. Primary peristaltic waves are triggered by swallowing. Secondary waves can be triggered by esophageal distension if material remains in the esophagus. These waves travel at about 4 centimeters per second, moving the bolus from pharynx to stomach in about 5 to 9 seconds. At the junction between esophagus and stomach, the lower esophageal sphincter, though not a distinct anatomical structure, functions as a physiological sphincter through tonic contraction of smooth muscle in the lower esophageal wall and support from the diaphragm where the esophagus passes through it. This sphincter normally remains contracted, preventing stomach contents from refluxing into the esophagus. It relaxes during swallowing to allow the bolus to enter the stomach, then contracts again. The stomach, a J-shaped organ in the upper abdomen, serves as a temporary storage reservoir and an initial site of protein digestion. Its capacity when empty is about 50 milliliters, but can expand to accommodate 1 to 1.5 liters comfortably and even more when necessary. This expandability allows consumption of large meals at intervals rather than requiring continuous eating. The stomach has several anatomical regions. The cardia is the small area surrounding the entrance from the esophagus. The fundus is the dome-shaped region to the left of and above the cardia. The body is the large central region. The pylorus is the funnel-shaped region leading to the small intestine, terminating in the pyloric sphincter that controls emptying into the duodenum. The stomach wall shows modifications of the basic 4-layer structure. The mucosa is deeply folded into rugae when the stomach is empty, allowing expansion when food enters. The epithelium is simple columnar with all surface cells secreting mucus that protects the lining from acid and pepsin. The epithelium invaginates to form millions of gastric pits, at the base of which lie gastric glands that produce the stomach's secretions. These gastric glands contain several cell types, each producing different secretions. Mucus cells, found throughout the stomach, secrete protective mucus. Parietal cells, concentrated in the body and fundus, secrete hydrochloric acid, an intrinsic factor, a protein necessary for vitamin B12 absorption later in the small intestine. Chief cells secrete pepsinogen, an inactive precursor of the protein-digesting enzyme pepsin. Enteroendocrine cells secrete various hormones, particularly gastrin from G cells in the pylorus. The muscularis externa of the stomach has 3 layers rather than the usual 2, with an additional oblique layer inside the circular layer. This 3rd layer enhances the stomach's ability to churn and mix contents. The coordinated contraction of these muscle layers produces both mixing movements that blend food with gastric secretions and propulsive movements that gradually empty the stomach. The stomach performs several important functions. It stores food, allowing intermittent eating rather than continuous consumption. It begins protein digestion through the action of pepsin in the acidic environment. It produces intrinsic factor essential for B12 absorption. Its muscular contractions mechanically break down food and mix it with secretions, creating a semi-liquid mixture called chyme. It regulates the rate at which chyme enters the small intestine, metering it out gradually to match the intestine's digestive and absorptive capacities. The stomach secretes about 2 liters of gastric juice daily. This secretion includes hydrochloric acid at a concentration that creates a pH of 1 to 3 in the stomach lumen, among the most acidic environments in the body. This extreme acidity serves multiple purposes. It denatures proteins, unfolding them for easier enzymatic digestion. It converts pepsinogen to pepsin. It kills most ingested microorganisms, providing a barrier against pathogens. It aids in dissolving certain minerals. Pepsin, the stomach's primary digestive enzyme, breaks peptide bonds within protein molecules, fragmenting proteins into smaller polypeptides. Pepsin works optimally in the acidic pH of the stomach and becomes inactive if pH rises. This explains why pepsin cannot digest proteins in the neutral environment of the small intestine and requires the stomach's acid for function. The stomach lining protects itself from its own acid and pepsin through several mechanisms. The epithelial cells secrete a thick mucus layer that coats the stomach lining. This mucus traps bicarbonate ions secreted by surface cells, creating a pH gradient from very acidic in the lumen to nearly neutral at the epithelial surface. The epithelial cells have tight junctions preventing acid from penetrating between cells. The cells replace themselves rapidly, with the entire stomach lining renewing every 3 to 5 days. If these protective mechanisms fail, the acid and pepsin can damage the lining, leading to inflammation or ulceration. Gastric emptying, the release of chyme from stomach to duodenum, is carefully regulated. Liquids begin emptying almost immediately, but solids can remain in the stomach for 2 to 6 hours depending on meal composition. The pyloric sphincter controls the rate of emptying through its degree of contraction. Factors that slow emptying include large meal volume, high fat content, high acidity, and high osmolarity in the duodenum. This regulation ensures that the small intestine receives chyme at a rate it can process effectively. The small intestine, extending from the pyloric sphincter to the ileocecal valve, is the primary site of digestion and absorption. Despite its name, it is the longest portion of the digestive tract, measuring about 6 to 7 meters in length. It coils extensively within the central and lower abdomen, fitting its considerable length into a compact space. The small intestine divides into 3 regions: the duodenum, jejunum, and ileum, and the duodenum, about 25 centimeters long, curves around the head of the pancreas in a C shape. This first section receives both chyme from the stomach and secretions from the pancreas and liver. The pancreatic duct and common bile duct join and enter the duodenum through the hepatopancreatic ampulla controlled by the sphincter of Oddi. The duodenum's strategic position at this confluence makes it a crucial site where multiple digestive secretions mix with chyme. The jejunum, about 2.5 meters long, occupies the upper left abdomen. The ileum, about 3.5 meters long, occupies the lower right abdomen and pelvis, eventually joining the large intestine at the ileocecal valve. No clear anatomical boundary separates jejunum and ileum, but they differ subtly in diameter, wall thickness, vascularization, and internal features. The small intestine wall shows extraordinary adaptations for increasing surface area, essential for its absorptive function. The mucosa forms circular folds called plicae circularis, particularly prominent in the duodenum and jejunum, which can triple surface area. The epithelium projects into millions of finger-like villi, each about 1 millimeter long, increasing surface area another tenfold. Each epithelial cell has a brush border of microvilli, microscopic projections of the cell membrane, increasing surface area another 20-fold. These 3 levels of folding, circular folds, villi, and microvilli, create a total absorptive surface area estimated at 200 to 300 square meters, roughly the size of a tennis court, packed within the volume of your abdomen. Each villus contains a central lacteal, a lymphatic capillary that absorbs fats, and a network of blood capillaries that absorb other nutrients. The epithelium covering the villi consists primarily of absorptive enterocytes, tall columnar cells specialized for nutrient uptake. Scattered among them are goblet cells that secrete mucus and enteroendocrine cells that produce hormones. Between the bases of villi are intestinal crypts where new epithelial cells continuously form, migrating up the villi to replace older cells that eventually slough off. The entire intestinal epithelium renews every 3 to 5 days, one of the most rapid turnover rates in the body. The submucosa of the duodenum contains submucosal glands that secrete alkaline mucus, helping to neutralize the acidic chyme entering from the stomach and protecting the duodenal lining. The muscularis externa maintains the typical 2-layer arrangement, producing the contractions that mix and propel contents. Digestion in the small intestine involves enzymes from 3 sources: the pancreas, the intestinal brush border, and to a lesser extent, the liver via bile. The pancreas, a gland lying behind the stomach, produces about 1.5 liters daily of pancreatic juice, an alkaline secretion rich in bicarbonate and digestive enzymes. The bicarbonate neutralizes stomach acid, raising the pH of chyme from acidic to slightly alkaline, creating optimal conditions for pancreatic and intestinal enzymes. Pancreatic enzymes include proteases that digest proteins, amylase that digests carbohydrates, lipase that digests fats, and nucleases that digest nucleic acids. The proteases, trypsin, chymotrypsin, and carboxypeptidase are secreted as inactive precursors and activated in the intestinal lumen, preventing them from digesting the pancreas itself. Trypsinogen is converted to trypsin by enterokinase, an enzyme in the intestinal brush border. Trypsin then activates the other pancreatic proteases, creating a controlled enzymatic cascade. Pancreatic amylase continues the starch digestion begun by salivary amylase, breaking starch into smaller chains and eventually into the disaccharide maltose. Pancreatic lipase breaks triglycerides into fatty acids and monoglycerides. Nucleases break down DNA and RNA into nucleotides. This enzymatic arsenal can digest essentially all types of macronutrients, though fiber, the indigestible carbohydrates, passes through largely unchanged. Bile, produced continuously by the liver and stored in the gallbladder, plays a crucial role in fat digestion, though it contains no enzymes. Bile consists of water, bile salts, cholesterol, lecithin, bile pigments, and electrolytes. Bile salts, derived from cholesterol, are amphipathic molecules with both water-soluble and fat-soluble regions. In the small intestine, bile salts emulsify fats, breaking large lipid droplets into smaller droplets with greater surface area for lipase action. Bile salts also form micelles, tiny aggregates that transport fatty acids and fat-soluble vitamins to the intestinal epithelium for absorption. The liver, the body's largest internal organ, weighing about 1.5 kilograms, produces about 1 liter of bile daily. Bile flows from the liver through hepatic ducts, some being stored and concentrated in the gallbladder. When fat enters the duodenum, hormones trigger the gallbladder to contract and the sphincter of Oddi to relax, releasing stored bile into the duodenum. After participating in fat digestion and absorption, most bile salts are reabsorbed in the terminal ileum and returned to the liver, a recycling process called enterohepatic circulation. The brush border of intestinal epithelial cells contains additional enzymes that complete digestion. Maltase, sucrase, and lactase break disaccharides into monosaccharides. Aminopeptidases break small peptides into amino acids. These brush border enzymes perform the final steps of digestion, producing molecules small enough for absorption. Absorption in the small intestine recovers nearly all nutrients, water, and electrolytes from the intestinal contents. Different nutrients use different absorption mechanisms. Monosaccharides and amino acids cross the epithelium through specific transport proteins, entering the blood capillaries within villi. Fats, being hydrophobic, diffuse through cell membranes as fatty acids and monoglycerides, are reassembled into triglycerides within epithelial cells, packaged with proteins into chylomicrons, and released into lacteals, entering the lymphatic system. Water follows osmotic gradients, moving from lumen to blood primarily through passive processes. About 8 to 10 liters of water enter the intestinal tract daily from food, drink, and various secretions. The small intestine absorbs about 7 liters, leaving 1 to 2 liters to enter the colon. This massive water movement is essential for maintaining the fluidity of intestinal contents necessary for enzymatic digestion. Vitamins, minerals, and electrolytes each have specific absorption mechanisms. Water-soluble vitamins generally use transporters. Fat-soluble vitamins A, D, E, and K dissolve in micelles and are absorbed with fats. Vitamin B12 requires intrinsic factor from the stomach and is absorbed specifically in the terminal ileum. Iron absorption is carefully regulated since the body has limited ability to excrete excess iron. Calcium absorption is enhanced by vitamin D and regulated according to body needs. By the time contents reach the end of the ileum, most nutrients and much of the water have been absorbed. What enters the large intestine is primarily water, electrolytes, some remaining nutrients, indigestible fiber, bacteria, and sloughed epithelial cells. The ileocecal valve controls passage from ileum to cecum, normally remaining closed but relaxing in response to eating and to distension of the ileum, allowing contents to enter the colon in spurts. As we complete this exploration of the foundational structures and functions from mouth through small intestine, notice how each region contributes specialized capabilities to the overall digestive process. The mouth mechanically fragments and mixes with saliva. The esophagus transports to the stomach. The stomach stores, acidifies, and begins protein digestion. The small intestine receives secretions from pancreas and liver, completes digestion, and accomplishes the bulk of absorption. Each region's structure reflects its function. Each component serves the whole. The journey of food through these passages represents transformation, a conversion from external matter to absorbable nutrients, from the complex structures of plants and animals to the simple molecules that will become you. This transformation proceeds through mechanical forces, chemical reactions, and selective transport, orchestrated by hormones, nerves, and cellular intelligence. The river of digestion flows ever onward, carrying nutrients from the world into your body, sustaining your existence meal by meal, day by day. We continue now from the small intestine's final segments into the broader channels and slower currents of the large intestine, where the character of digestion changes fundamentally. The remaining contents, already depleted of most nutrients, enter a realm where water reclamation becomes paramount, where resident microorganisms outnumber human cells, where matter transitions from liquid to solid, as it approaches its final departure from the body. This is not diminished importance, but different purpose, completing the digestive cycle that began with that first bite of food. The large intestine, also called the colon, frames the abdominal cavity in an inverted U shape, extending about 1.5 meters in length, but with a diameter roughly twice that of the small intestine, hence its name. It begins with the cecum in the lower right abdomen, ascends along the right side as the ascending colon, crosses the upper abdomen as the transverse colon, descends along the left side as the descending colon, forms an S-curve as the sigmoid colon, and terminates as the rectum and anal canal. The cecum, a blind pouch below the ileocecal valve, marks the beginning of the large intestine. Attached to the cecum is the appendix, a narrow tubular structure about 9 centimeters long, whose function in humans remains somewhat mysterious. The appendix contains abundant lymphoid tissue and may play a role in immune function or serve as a reservoir for beneficial gut bacteria. Its narrow lumen makes it vulnerable to obstruction and inflammation, causing appendicitis. The large intestine wall differs from the small intestine in several ways. The mucosa lacks villi, presenting a relatively smooth surface except for abundant crypts where epithelial cells produce copious mucus. This mucus lubricates the passage of increasingly solid contents and protects the lining from mechanical damage and bacterial products. The epithelium consists primarily of absorptive cells specialized for water and electrolyte uptake, and numerous goblet cells secreting protective mucus. The muscularis externa shows a unique modification. Rather than forming a continuous outer longitudinal layer, the longitudinal muscle concentrates into 3 bands called tenae coli that run the length of the colon. Contraction of these bands gathers the colon into a series of pouches called haustra, visible on the outer surface. This architecture facilitates the mixing and propulsive movements characteristic of colonic motility. The primary function of the large intestine is water and electrolyte absorption. Of the 1 to 2 liters of chyme entering from the small intestine daily, the colon absorbs about 90%, leaving only 100 to 200 milliliters to be excreted in feces. This water reclamation is crucial for maintaining fluid balance. Sodium is actively transported from lumen to blood, and water follows osmotically. Potassium and bicarbonate are secreted into the lumen in exchange. This absorption concentrates the intestinal contents, transforming the liquid chyme into semi-solid, then solid feces. The colon also harbors the gut microbiota, a vast community of microorganisms living in mutualistic relationship with the human host. While bacteria exist throughout the digestive tract, their density increases dramatically in the colon, reaching concentrations of 1 trillion cells per gram of content. These microorganisms, predominantly bacteria, but also including archaea, fungi, and viruses, outnumber human cells in the body and collectively contain more genes than the human genome. The gut microbiota performs numerous beneficial functions. Bacteria ferment indigestible carbohydrates, particularly fiber, that escaped digestion in the small intestine. This fermentation produces short-chain fatty acids, acetate, propionate, and butyrate, which colonic epithelial cells absorb and use as energy sources. Butyrate particularly nourishes colonocytes and helps maintain intestinal barrier function. The microbiota synthesizes certain vitamins, particularly vitamin K, and some B vitamins, contributing to nutritional status. These organisms also metabolize bile acids, influencing cholesterol metabolism. The immune relationship between host and microbiota is complex and finely balanced. The intestinal immune system recognizes commensal bacteria as non-threatening despite their presence in enormous numbers. This tolerance develops through intricate molecular signaling between bacteria and immune cells. The microbiota, in turn, helps educate and shape the immune system, particularly during early life. Germ-free animals raised without any microorganisms show impaired immune development, demonstrating the microbiota's crucial role in immune maturation. The composition of the gut microbiota varies among individuals and changes throughout life. Infant microbiota differs from adult microbiota. Diet profoundly influences microbial populations, with different bacterial species specializing in fermenting different types of fiber. Antibiotic use can dramatically alter the microbiota, killing susceptible species and potentially allowing resistant organisms or pathogens to proliferate. The microbiota's influence extends beyond the gut, affecting metabolism, immune function, and even behavior and cognition through the gut-brain axis, making it a subject of intense research interest. Colonic motility patterns differ from those of the small intestine. Haustral contractions are slow-mixing movements that shift contents from one haustrum to another, exposing material to the absorptive surface. These contractions occur every 30 minutes or so. Mass movements are powerful peristaltic waves that propel contents forward over considerable distances, typically occurring a few times daily, often triggered by eating through the gastrocolic reflex. These mass movements push fecal material into the rectum. The rectum, normally empty, serves as a temporary holding chamber. When mass movements deliver feces to the rectal walls distend, triggering the defecation reflex. Stretch receptors in the rectal wall send signals to the spinal cord, initiating reflexive relaxation of the internal anal sphincter, a smooth muscle under involuntary control. This creates the sensation of needing to defecate. The external anal sphincter, skeletal muscle under voluntary control, can maintain continence by remaining contracted or can relax to allow defecation. Defecation involves coordinated actions. The individual finds an appropriate time and place, assumes a squatting or sitting position, and voluntarily relaxes the external anal sphincter. The Valsalva maneuver, closing the glottis and contracting abdominal muscles, increases intra-abdominal pressure, compressing the colon. Simultaneously, rectal contractions push feces toward the anus. The puborectalis muscle, which normally maintains a bend in the rectum that aids continence, relaxes to straighten the pathway. Feces pass through the anal canal and are expelled. The feces consist of water, indigestible fiber, sloughed epithelial cells, bacteria both living and dead, bile pigments that give feces their characteristic color, and small amounts of unabsorbed nutrients. The brown color comes from stercobilin, a breakdown product of bilirubin from degraded red blood cells. The odor comes from bacterial metabolites, particularly sulfur-containing compounds and indole. Consistency ranges from hard and formed when transit time is prolonged and water absorption maximal to loose or liquid when transit is rapid or water absorption impaired. Normal bowel movement frequency varies widely among healthy individuals, from 3 times daily to 3 times weekly. Constipation refers to difficult or infrequent defecation, typically from prolonged transit time, allowing excessive water absorption and hard stool formation. Diarrhea refers to frequent loose or watery stools, typically from rapid transit limiting water absorption. from increased secretion of water into the lumen or from osmotic substances drawing water into the lumen. Both represent disruptions of the colon's normal water handling function. Having traced the anatomical journey from mouth through colon to rectum, we now shift perspective to examine the regulatory systems that coordinate digestive activities. Digestion is not a passive flow, but an actively regulated process, with secretions, motility, and absorption all controlled to match current needs. This regulation operates at multiple levels, neural, hormonal, and local, creating an integrated control network. The autonomic nervous system influences digestive function through both sympathetic and parasympathetic divisions. Parasympathetic stimulation, primarily through the vagus nerve, generally promotes digestive activities. It increases salivary secretion, gastric secretion, pancreatic secretion, and bile release. It enhances motility in the stomach and intestines. The vagus nerve carries signals from the brain to the digestive organs and sensory information from the organs back to the brain, creating bidirectional communication. Sympathetic stimulation, arising from thoracic and lumbar spinal segments, generally inhibits digestion. It decreases secretions and motility, redirecting blood flow away from digestive organs towards skeletal muscles and heart. This makes physiological sense during stress or physical activity when energy is needed for fight or flight rather than for digestion. The sympathetic nerves also carry pain signals from the digestive organs to the central nervous system. The enteric nervous system, introduced earlier, deserves deeper consideration as the digestive tract's intrinsic control system. This network of neurons embedded in the gut wall can coordinate digestive functions independently, though it normally communicates extensively with the central nervous system. The enteric nervous system contains sensory neurons that detect chemical and mechanical stimuli in the gut lumen, interneurons that process information, and motor neurons that control smooth muscle, secretory cells, and blood vessels. The myenteric plexus, located between the longitudinal and circular muscle layers, primarily controls motility. The submucosal plexus primarily controls secretion and local blood flow. These plexuses connect with each other and with the autonomic nervous system, creating a sophisticated control network. The enteric nervous system uses numerous neurotransmitters, including many also found in the central nervous system, supporting the second brain designation. Reflexes coordinated by the enteric nervous system include the peristaltic reflex, where distension at one location triggers contraction above and relaxation below, propelling contents forward. The gastrocolic reflex, where eating triggers increased colonic motility, helps clear the colon for incoming material. The enterogastric reflex, where distension or chemical irritation in the intestine inhibits gastric emptying, prevents overloading the intestine. These reflexes operate largely automatically, adjusting digestive activities to current conditions. Gastrointestinal hormones provide another layer of regulation, coordinating activities among different digestive organs. These hormones are produced by enteroendocrine cells scattered throughout the digestive epithelium. The cells sense nutrients or other stimuli in the gut lumen and release hormones into the bloodstream, which carries them to target organs throughout the body. Gastrin, produced by G cells in the stomach pylorus and duodenum, stimulates gastric acid secretion, promotes gastric motility, and stimulates growth of gastric mucosa. Its release is triggered by protein in the stomach, by distension, and by parasympathetic stimulation. Gastrin levels rise during meals, supporting the increased acid secretion needed for protein digestion. When stomach pH drops below 2, gastrin release is inhibited, providing negative feedback that prevents excessive acidity. Secretin, produced by S cells in the duodenum, is released in response to acidic chyme entering from the stomach. Secretin stimulates the pancreas to secrete bicarbonate-rich fluid that neutralizes the acid, protecting the duodenal lining and creating the pH optimal for pancreatic enzymes. Secretin also inhibits gastric acid secretion and slows gastric emptying, preventing further acid delivery until the duodenum has neutralized what is already there. Cholecystokinin, abbreviated CCK, is produced by I cells in the duodenum and jejunum in response to fatty acids and amino acids. CCK stimulates the gallbladder to contract, releasing stored bile for fat emulsification. It stimulates pancreatic enzyme secretion, providing the enzymes needed to digest the arriving nutrients. It slows gastric emptying, ensuring the small intestine has adequate time to process the meal. CCK also acts in the brain to promote satiety, the feeling of fullness that reduces further food intake. Gastric inhibitory peptide, also called glucose-dependent insulinotropic peptide or GIP, is released by K cells in the duodenum and jejunum in response to glucose and fats. GIP inhibits gastric acid secretion and slows gastric emptying. It also stimulates insulin release from the pancreas, preparing the body to handle the absorbed glucose. This demonstrates how gut hormones coordinate not just digestion, but also metabolic responses to nutrients. Motilin, produced by M cells in the duodenum, stimulates gastric and intestinal motility. Motilin levels rise during fasting, triggering the migrating motor complex, a pattern of strong contractions that sweeps through the stomach and small intestine every 90 to 120 minutes between meals. This intestinal housekeeper clears residual contents and bacteria, preventing bacterial overgrowth. Eating suppresses motilin and the migrating motor complex, replacing it with the fed motility pattern. These hormones and others create a chemical communication network that coordinates digestive activities with nutrient arrival, matching secretions to digestive needs, adjusting motility to optimize digestion and absorption, and linking digestive processes to broader metabolic regulation. The hormonal system works in concert with neural control, each complementing the other's strengths. Hormones providing slower but sustained effects that can coordinate distant organs, nerves providing rapid point-to-point communication. The regulation of eating itself involves complex interactions between digestive signals and brain centers, controlling appetite and satiety. Hunger, the drive to eat, and satiety, the feeling of fullness that terminates eating, involve both short-term signals related to individual meals and long-term signals related to energy stores. Short-term regulation begins before eating with the cephalic phase, where the sight, smell, thought, or taste of food triggers anticipatory responses. Saliva secretion increases, gastric secretion increases, insulin secretion may begin. These preparatory responses prime the digestive system for the incoming meal. The cephalic phase demonstrates how higher brain centers influence digestive function. During eating, gastric distension sends signals through the vagus nerve to the brainstem, contributing to satiety. The rate of gastric emptying influences how quickly fullness develops and how long it lasts. Nutrients in the small intestine trigger release of satiety hormones, including CCK, peptide YY, and glucagon-like peptide 1. These hormones act both peripherally to slow digestion and centrally in the brain to reduce appetite. The combination of gastric distension and intestinal nutrient signals creates the feeling of fullness that ends a meal. Long-term regulation involves leptin, a hormone produced by adipose tissue in proportion to fat stores. Leptin acts in the hypothalamus to reduce appetite and increase energy expenditure when fat stores are sufficient. Ghrelin, produced primarily by the stomach, increases before meals and stimulates appetite. The balance between leptin and ghrelin, along with insulin and other metabolic hormones, adjusts food intake to match energy expenditure and maintain relatively stable body weight over time. The gut-brain axis encompasses these appetite-regulating pathways, plus broader bidirectional communication between the digestive system and the brain. Gut signals influence mood, stress responses, and cognitive function. Brain states influence gut motility, secretion, and sensitivity. The vagus nerve serves as a major communication channel in both directions. Gut hormones can cross the blood-brain barrier or signal through vagal afferents. The gut microbiota produces neurotransmitters and other signaling molecules that can influence brain function. This gut-brain communication helps explain phenomena like stress-induced changes in digestion. Anxiety can increase acid secretion, alter motility causing diarrhea or constipation, and lower the threshold for abdominal pain. Chronic stress can exacerbate functional digestive disorders. Conversely, digestive problems can influence mood and cognitive function, creating bidirectional relationships between gut health and mental health. Now we examine the liver more thoroughly, this remarkable organ whose functions extend far beyond bile production. The liver, weighing about 1.5 kilograms and located in the upper right abdomen beneath the diaphragm, is the body's largest internal organ and performs hundreds of functions essential for life. It is unique among organs in receiving blood from two sources. The hepatic artery provides oxygen-rich blood from the heart, while the hepatic portal vein brings nutrient-rich blood from the digestive organs. The liver's microarchitecture consists of hepatic lobules, roughly hexagonal structures about 1 millimeter across. At each lobule's center is a central vein. At the periphery are portal triads, containing branches of the hepatic artery, hepatic portal vein, and bile duct. Between the central vein and portal triads run sinusoids, specialized capillaries lined with fenestrated endothelium that allows easy exchange between blood and hepatocytes. Hepatocytes, the liver's main functional cells, are arranged in plates 1 or 2 cells thick that radiate from the central vein toward the periphery. Blood flows from portal triads through the sinusoids toward the central vein, exposing hepatocytes to blood in a sequential manner. Hepatocytes near portal triads encounter blood richest in oxygen and nutrients. Those near the central vein see blood already partially processed by upstream hepatocytes. This zonation creates functional differences, with different hepatocytes specializing in different metabolic tasks. The liver's metabolic functions are extensive. It regulates blood glucose levels, storing excess glucose as glycogen after meals and breaking down glycogen to release glucose during fasting. When glycogen stores are depleted, the liver performs gluconeogenesis, synthesizing new glucose from amino acids and other precursors. This glucose regulation is crucial for maintaining the constant blood glucose levels that the brain requires. The liver synthesizes most plasma proteins, including albumin, which maintains blood osmotic pressure, clotting factors essential for hemostasis, and transport proteins that carry hormones, vitamins, and other molecules through the bloodstream. The liver produces cholesterol and lipoproteins, that transport lipids through the blood. It metabolizes amino acids, converting excess amino acids into glucose or fat, and producing urea from the ammonia generated by amino acid breakdown. Detoxification represents another vital hepatic function. The liver processes drugs, alcohol, and other foreign compounds, typically converting them to more water-soluble forms that can be excreted in bile or urine. The cytochrome P450 enzyme system in hepatocytes performs many of these conversions. The liver also processes and excretes bilirubin, the breakdown product of hemoglobin from aged red blood cells. Bilirubin gives bile its characteristic color and, when excreted in feces, creates the normal brown color. The liver stores various nutrients beyond glycogen. It stores fat-soluble vitamins A, D, E, and K, along with vitamin B12 and minerals like iron and copper. These stores can be mobilized when dietary intake is inadequate, providing a buffer against nutritional deficiencies. Bile production, while just one of many liver functions, is essential for digestion. Hepatocytes continuously produce bile, secreting it into tiny bile canaliculi between adjacent hepatocytes. The canaliculi merge into progressively larger bile ducts that eventually exit the liver as the common hepatic duct. This duct joins the cystic duct from the gallbladder to form the common bile duct that delivers bile to the duodenum. The gallbladder, a pear-shaped sac about 7 to 10 centimeters long, stores and concentrates bile between meals. The gallbladder epithelium absorbs water and electrolytes, concentrating bile 5 to 20-fold. This concentration increases bile's effectiveness at emulsifying fats. When fat enters the duodenum and CCK is released, the gallbladder contracts rhythmically, the sphincter of Oddi relaxes, and concentrated bile flows into the duodenum. Gallstones can form when bile components, particularly cholesterol or bilirubin, precipitate and crystallize. If stones block the cystic duct, they cause gallbladder pain. If they enter and obstruct the common bile duct, they can cause jaundice by preventing bile excretion and backing up bilirubin into the blood. Most gallstones remain asymptomatic, but symptomatic stones may require removal of the gallbladder, after which bile flows directly from liver to duodenum. The pancreas deserves fuller exploration as well, this dual-function gland serving both digestive and endocrine roles. The pancreas, about 15 centimeters long, lies retroperitoneally, behind the peritoneum, extending from the curve of the duodenum across to the spleen. Its head nestles into the duodenal C-curve. Its body extends horizontally, and its tail reaches the spleen. About 99% of the pancreas consists of acinar cells organized into clusters called acini. These exocrine cells produce and secrete the pancreatic digestive enzymes. The acini drain into a branching duct system that eventually merges into the main pancreatic duct running the length of the gland. This duct typically joins the common bile duct just before entering the duodenum, though anatomical variations exist. The remaining 1% of pancreatic tissue consists of islets of Langerhans, small clusters of endocrine cells scattered throughout the gland. These islets produce hormones, particularly insulin and glucagon, that regulate blood glucose levels. Insulin, produced by beta cells, lowers blood glucose by promoting cellular uptake and storage. Glucagon, produced by alpha cells, raises blood glucose by stimulating glycogen breakdown and gluconeogenesis in the liver. While these endocrine functions are crucial, they are separate from the digestive functions we focus on here. Pancreatic secretion is carefully regulated to match digestive needs. During the cephalic phase, vagal stimulation triggers modest enzyme secretion. When acidic chyme enters the duodenum, secretin triggers bicarbonate secretion to neutralize the acid. When fats and proteins enter the duodenum, CCK triggers enzyme secretion. The combined neural and hormonal signals can increase pancreatic secretion 20-fold during digestion compared to basal rates. The pancreatic enzymes are so powerful that they could digest the pancreas itself if activated prematurely. The proteases are therefore secreted as inactive zymogens that become activated only after reaching the duodenal lumen. Trypsinogen is activated to trypsin by enterokinase from the intestinal brush border. Trypsin then activates the other proteases and also activates more trypsinogen in a positive feedback cascade. The pancreas protects itself by secreting a trypsin inhibitor that inactivates any trypsin that might form prematurely. If this protective system fails and pancreatic enzymes become active within the pancreas, acute pancreatitis can result, with the enzymes digesting pancreatic tissue and causing severe inflammation. This painful and potentially serious condition demonstrates the power of digestive enzymes and the importance of the mechanisms that normally confine their activity to the gut lumen. The integration of liver, gallbladder, and pancreatic functions with intestinal digestion demonstrates the coordinated nature of the digestive process. The liver produces bile containing salts needed for fat digestion. The gallbladder stores and concentrates this bile. The pancreas produces enzymes for digesting all major nutrients, plus bicarbonate for neutralizing acid. The duodenum receives these secretions exactly when needed, triggered by hormones released in response to the nutrients requiring digestion. The small intestine completes digestion with brush border enzymes and accomplishes absorption through its specialized epithelium. Each organ contributes essential components at precisely the right time and place. This coordination extends to the circulation as well. The hepatic portal system, unique among venous systems, carries blood from the digestive organs directly to the liver before returning to the heart. Capillaries in the intestinal villi absorb nutrients into venules that merge into larger veins. These eventually form the superior mesenteric vein from the small intestine and part of the colon, and the inferior mesenteric vein from the rest of the colon. These veins join with the splenic vein to form the hepatic portal vein. The portal vein carries nutrient-rich blood from the intestines to the liver, allowing the liver to process absorbed nutrients before they enter the general circulation. The liver can remove excess glucose and store it as glycogen, preventing blood glucose from spiking too high after meals. It can modify absorbed nutrients, converting some amino acids to others, synthesizing lipoproteins to transport fats, and beginning the metabolism of various compounds. This first-pass effect through the liver is crucial for metabolic regulation and detoxification. From the liver, blood flows through hepatic sinusoids, past hepatocytes that extract and process nutrients, then collects in central veins. Central veins merge into hepatic veins that drain into the inferior vena cava, returning blood to the heart. This blood, now modified by hepatic processing, mixes with blood from the rest of the body and is pumped to the lungs and back to the left heart, then distributed throughout the body via the systemic arterial system. The immune functions of the gastrointestinal system deserve deeper exploration. The gut faces an enormous immune challenge, constantly exposed to food antigens, commensal bacteria, and potential pathogens. It accomplishes the remarkable feat of mounting aggressive responses to threats while tolerating harmless or beneficial exposures, a discrimination essential for both protection and digestive function. The gut-associated lymphoid tissue, abbreviated GALT, represents the body's largest immune compartment. Peyer's patches, aggregates of lymphoid follicles in the small intestine, sample luminal contents through specialized M cells that transport antigens from the lumen to underlying immune cells. Isolated lymphoid follicles scattered throughout the intestinal wall perform similar functions. The lamina propria contains numerous immune cells, including macrophages, dendritic cells, and lymphocytes that continuously monitor the intestinal environment. The intestinal epithelium itself participates in immune function. Tight junctions between epithelial cells create a physical barrier preventing most microorganisms and large molecules from crossing between cells. Epithelial cells produce antimicrobial peptides that kill bacteria attempting to penetrate the mucus layer. They express pattern recognition receptors that detect bacterial components and trigger appropriate responses. The mucus layer provides another defensive barrier, keeping bacteria at a distance from the epithelium. The inner mucus layer is dense and largely devoid of bacteria. The outer layer is looser and harbors commensal bacteria. This spatial segregation allows peaceful coexistence with the microbiota while preventing bacterial invasion of the tissue. Secretory IgA, the predominant antibody in the gut lumen, is produced by plasma cells in the lamina propria and transported across the epithelium. This antibody binds to bacteria and toxins, neutralizing threats without triggering inflammation. The high concentrations of IgA in the gut represent continuous immune surveillance without the tissue damage that other antibody classes might cause. The development of oral tolerance, the immune system's learned acceptance of harmless food antigens, prevents food allergies. Regulatory T cells in the gut help establish and maintain this tolerance. When this process fails, food allergies develop, where the immune system inappropriately attacks food proteins, causing symptoms ranging from mild discomfort to life-threatening anaphylaxis. The microbiota profoundly influences immune development and function. Germ-free animals have underdeveloped immune systems that mature when colonized with bacteria. Specific bacterial species can promote different types of immune responses. The microbiota helps educate the immune system to distinguish friend from foe, shapes the development of immune tissues, and influences susceptibility to inflammatory and autoimmune conditions. As we conclude this exploration of regulatory systems, circulation, and immunity, we recognize the gastrointestinal system's extraordinary sophistication. It is not merely a digestive tube, but an integrated system performing multiple vital functions: digestion, absorption, immune surveillance, hormone production, microbial housing. The coordination among these functions, the intricate regulatory networks, and the complex relationships with other body systems reveal the digestive tract as a masterpiece of biological organization. The river of digestion flows through varied landscapes, each section uniquely suited to its role. The coordination is so seamless that we can eat a meal and trust that the proper enzymes will be secreted, the proper absorption will occur, the proper immune responses will engage, all without conscious direction. This automation, this reliable function, this quiet intelligence supports your existence, meal after meal, allowing you to nourish yourself and sustain life. As we move deeper into understanding the gastrointestinal system, we begin to see it not as a collection of separate organs, but as an integrated whole, a system whose parts communicate constantly, whose functions interweave seamlessly, whose health depends on the harmonious interaction of structure, chemistry, microbes, and regulation. The digestive process we have traced from mouth to colon represents just one dimension of a multifaceted system that touches nearly every aspect of human physiology. Consider how eating a single meal initiates cascades of responses throughout the body. Before the first bite reaches your mouth, the cephalic phase has already begun. The sight of food activates visual cortex. The aroma stimulates olfactory receptors. These sensory signals reach the brain, particularly the hypothalamus and limbic system, triggering anticipatory responses. Salivary glands increase secretion. The stomach begins producing acid even before food arrives. The pancreas prepares to release enzymes. These preparatory changes demonstrate how the brain and digestive system communicate bidirectionally, how thought and sensation influence physiology. As food enters the mouth, taste receptors on the tongue detect sweet, sour, salty, bitter, and umami flavors. These are not merely sensations, but information-gathering processes. Sweet taste indicates energy-rich carbohydrates. Umami signals protein-containing foods. Bitter taste warns of potential toxins. The information travels through cranial nerves to the brainstem and onward to the gustatory cortex, creating the conscious experience of taste, while simultaneously influencing digestive secretions and hormones. The texture of food, detected by mechanoreceptors in the mouth, influences chewing patterns. Tough, fibrous meat requires prolonged grinding by molars. Soft bread compresses easily. Liquid requires no chewing but triggers immediate swallowing reflexes. The jaw muscles, tongue, and cheeks coordinate unconsciously, adapting to each bite's properties. This sensory-motor coordination happens so automatically that you can eat while conversing or reading, with the eating mechanics proceeding in the background of consciousness. Temperature receptors detect whether food is hot or cold, adjusting secretions and protecting against thermal damage. Very hot foods might burn the oral mucosa if consumed too quickly. Very cold foods can temporarily numb taste receptors. The comfortable temperature range for eating reflects the conditions under which digestive enzymes function optimally and under which the epithelium remains healthy. Swallowing that seemingly simple act requires exquisite coordination of over 25 pairs of muscles. The tongue propels the bolus backward. The soft palate elevates, sealing the nasopharynx. The larynx rises, the epiglottis folds down, the vocal cords close, all protecting the airway. The upper esophageal sphincter relaxes at precisely the right moment. This choreography happens in about 1 second, coordinated by the swallowing center in the medulla. The fact that we rarely choke or aspirate during thousands of swallows demonstrates the reliability of this neural program. As the bolus travels down the esophagus, its movement is tracked through sensory neurons detecting stretch. If the bolus fails to reach the stomach, secondary peristaltic waves are triggered automatically to clear the esophagus. The lower esophageal sphincter, maintaining baseline tone through both muscular contraction and support from the diaphragmatic crura, must relax at exactly the right moment to admit the bolus while preventing reflux of acidic stomach contents. Gastroesophageal reflux disease, commonly known as GERD or heartburn, occurs when the sphincter function becomes impaired. Acid from the stomach refluxes into the esophagus, where the epithelium, unlike the stomach lining, lacks protection against acid. The acid causes irritation and inflammation. producing the burning sensation felt behind the sternum. Chronic reflux can lead to changes in the esophageal epithelium, where normal stratified squamous epithelium transforms into columnar epithelium, more resistant to acid but potentially prone to malignant transformation. In the stomach, the arrival of food triggers the gastric phase of digestion. Distension of the stomach wall activates stretch receptors that signal through the vagus nerve and through local reflexes. Proteins in food stimulate G cells to release gastrin. The gastrin enters the bloodstream, circulates to parietal cells, and stimulates acid secretion. This creates a positive feedback loop where protein in the stomach promotes acid secretion that helps digest that protein. The stomach's acid secretion demonstrates remarkable cellular specialization. Parietal cells contain an elaborate network of secretory canaliculi lined with millions of proton pumps. These pumps actively transport hydrogen ions from inside the cell into the canaliculus against a tremendous concentration gradient. The pH inside the cell is about 7, while the pH in the canaliculus and stomach lumen can drop to 1 or 2, representing a more than 1 million-fold concentration difference. This active transport requires substantial energy, supplied by abundant mitochondria within parietal cells. The hydrogen ions combine with chloride ions to form hydrochloric acid. The bicarbonate ions generated as a byproduct of acid secretion enter the bloodstream, slightly raising blood pH. The so-called alkaline tide that can be detected in venous blood draining the stomach during active digestion. This elegant arrangement allows the stomach to secrete very strong acid without disturbing overall acid-base balance. Pepsinogen, secreted by chief cells as an inactive zymogen, is activated to pepsin by the acidic pH. Pepsin then catalyzes the conversion of more pepsinogen to pepsin, creating autocatalytic activation. Pepsin breaks internal peptide bonds in proteins, fragmenting them into smaller polypeptides. Unlike the pancreatic proteases that will act later, pepsin shows relatively low specificity, cleaving at many different sites. This initial fragmentation makes proteins more accessible to the highly specific pancreatic proteases that complete digestion in the small intestine. The stomach's churning movements, generated by the 3 muscle layers, mechanically break down food while mixing it thoroughly with gastric secretions. The rhythm of these contractions varies in different regions. The fundus remains relatively quiescent, serving as a reservoir. The body exhibits gentle mixing waves. The pylorus shows vigorous contractions that grind solid particles and force liquid chyme through the pyloric sphincter while retarding solid particles, a process called sieving. Only particles smaller than about 2 millimeters can pass into the duodenum, ensuring adequate mechanical breakdown before intestinal digestion. The rate of gastric emptying is precisely controlled by multiple factors. The duodenum sends inhibitory signals when it becomes distended, when its pH drops too low, or when it detects fats, proteins, or hyperosmotic solutions. These signals, mediated by hormones like secretin and CCK, and by neural reflexes, slow gastric emptying, preventing the intestine from being overwhelmed. This creates a negative feedback system where the intestine regulates its own input rate. Carbohydrates empty fastest from the stomach, followed by proteins, with fats emptying most slowly. A meal rich in fat might remain in the stomach for 6 hours or more, while a carbohydrate-rich meal might empty in 2 to 3 hours. This differential emptying explains why fatty meals create prolonged satiety. The stomach remains full longer, continuing to send satiety signals to the brain. As chyme enters the duodenum, it encounters the alkaline secretions from the pancreas and the bile from the liver. The rapid pH shift from 1 or 2 in the stomach to 7 or 8 in the duodenum represents a dramatic environmental change occurring over just a few centimeters. This pH change serves multiple purposes. It inactivates pepsin, protects the duodenal mucosa from acid damage, and creates the optimal pH for pancreatic and brush border enzymes. The emulsification of fats by bile salts transforms large lipid droplets into millions of smaller droplets with vastly increased surface area. This physical process is essential because lipases are water-soluble enzymes that can only act at the lipid-water interface. By increasing this interface area thousands-fold, emulsification accelerates fat digestion enormously. Without bile, fat digestion would be severely impaired, and much fat would pass through unabsorbed, appearing in feces as steatorrhea. The micelles formed by bile salts serve as vehicles transporting fatty acids, monoglycerides, cholesterol, and fat-soluble vitamins to the intestinal epithelium. These micelles are small enough to diffuse through the unstirred water layer adjacent to the epithelial surface, something larger lipid droplets cannot do. At the epithelial surface, the lipid components dissociate from the micelle and diffuse across the cell membrane, while the bile salts remain in the lumen to be reabsorbed later in the ileum. Inside the enterocytes, fatty acids and monoglycerides are reassembled into triglycerides in the smooth endoplasmic reticulum. These triglycerides, along with cholesterol and fat-soluble vitamins, are packaged with proteins into large lipoprotein particles called chylomicrons. The chylomicrons are too large to enter blood capillaries, so they enter the central lacteal within each villus, entering the lymphatic system instead. The lymph eventually drains into the bloodstream at the junction of the left subclavian and internal jugular veins, allowing absorbed fats to bypass the liver initially. This lymphatic route for fat absorption explains why the lymph draining the intestine during fat absorption appears milky white, a condition called chyle. It also explains why fat-soluble vitamins and fat-containing drugs follow this pathway, affecting their metabolism and distribution. Some drugs are formulated to be taken with fatty meals specifically to enhance their absorption through this route. Water-soluble nutrients, monosaccharides, amino acids, water-soluble vitamins, minerals are absorbed across the enterocytes and enter the blood capillaries within the villi. This blood flows into venules that merge into the superior mesenteric vein, carrying absorbed nutrients directly to the liver through the hepatic portal system. This first pass through the liver allows immediate processing of absorbed nutrients. The liver's metabolic capabilities transform absorbed nutrients to meet body needs. Excess glucose is stored as glycogen or converted to fat. Amino acids are used for protein synthesis or converted to glucose or fat. The liver synthesizes plasma proteins, clotting factors, and other essential molecules. It packages triglycerides and cholesterol into lipoproteins for transport to other tissues. It begins metabolizing drugs and toxins that were absorbed from the gut. This central processing role makes the liver essential for maintaining metabolic homeostasis. The concept of bioavailability, the fraction of an absorbed substance that reaches the systemic circulation in active form, depends heavily on hepatic first-pass metabolism. Some drugs are extensively metabolized during this first pass through the liver, reducing bioavailability. Prodrugs, Inactive compounds that require hepatic conversion to active forms depend on this first-pass metabolism for activation. Understanding these hepatic processes is crucial for pharmacology and nutrition. The enterohepatic circulation of bile salts represents an elegant recycling system. Bile salts are secreted from the liver into bile, stored in the gallbladder, released into the duodenum, participate in fat emulsification and micelle formation throughout the small intestine, and are then actively reabsorbed by specific transporters in the terminal ileum. This reabsorbed bile salt pool returns through the portal blood to the liver, where it is extracted and secreted again into bile. This cycle repeats 6 to 8 times per day, meaning the total bile salt pool of about 3 grams participates in digesting perhaps 18 to 24 grams worth of fat absorption. Without this recycling, the liver would need to synthesize far more bile salts from cholesterol, and bile salt availability would limit fat digestion. The efficiency of enterohepatic circulation allows adequate fat digestion despite the relatively small bile salt pool. Disruption of this cycle, as occurs with ileal disease or resection, can cause bile salt malabsorption. The bile salts then reach the colon, where they irritate the colonic mucosa and stimulate water secretion, causing diarrhea. Additionally, the liver must synthesize more bile salts to replace those lost in feces, drawing down hepatic cholesterol stores and potentially leading to increased cholesterol synthesis. The absorption of specific nutrients reveals the intestinal epithelium's remarkable selectivity and specialization. Vitamin B12 absorption demonstrates this complexity. Dietary B12 is initially bound to proteins. Stomach acid and pepsin free the vitamin, which then binds to R proteins from saliva. In the duodenum, pancreatic enzymes degrade the R proteins, freeing B12 to bind to intrinsic factor from stomach parietal cells. This B12 intrinsic factor complex travels to the terminal ileum, where specific receptors recognize and internalize it. The complexity of this pathway explains why B12 deficiency can result from stomach problems affecting intrinsic factor production, pancreatic insufficiency, or ileal disease. Iron absorption is tightly regulated because the body has limited ability to excrete excess iron, yet inadequate iron causes anemia. Enterocytes in the duodenum take up dietary iron through specific transporters. The iron can either be stored within the enterocyte bound to ferritin or transferred across the basolateral membrane into the bloodstream via ferroportin. When body iron stores are adequate, more absorbed iron is retained in enterocytes and lost when those cells slough off. When iron is needed, more is transferred to blood. This cellular mechanism allows the intestine to regulate iron absorption according to body needs. Calcium absorption, influenced by vitamin D and parathyroid hormone, occurs throughout the small intestine, but primarily in the duodenum and jejunum. Active transport mechanisms move calcium against concentration gradients when dietary intake is low. Passive diffusion contributes when luminal calcium concentration is high. Vitamin D increases expression of calcium-binding proteins and transporters, enhancing absorption. This regulation ensures adequate calcium for bone formation and other calcium-dependent processes while preventing excessive accumulation. The absorption of water and electrolytes represents quantitatively the largest absorptive task. About 8 to 10 liters of fluid enter the small intestine daily, 2 liters from diet and the rest from salivary, gastric, biliary, pancreatic, and intestinal secretions. The small intestine absorbs about 7 liters, leaving 1 to 2 liters to enter the colon, which absorbs another 900 milliliters or so, leaving only 100 to 200 milliliters excreted in feces. This massive fluid absorption is crucial for maintaining hydration. Sodium absorption drives water absorption through osmotic effects. In the jejunum, sodium is absorbed along with glucose and amino acids through coupled transporters, the sodium-glucose cotransporter and sodium amino acid cotransporters. This coupling explains why oral rehydration solutions containing glucose and salt are so effective for treating dehydration from diarrhea. The glucose enhances sodium absorption, and water follows osmotically. In the ileum and colon, sodium is absorbed through sodium-hydrogen exchangers and epithelial sodium channels. Chloride follows through both paracellular and transcellular pathways. Bicarbonate is secreted into the lumen in exchange for chloride, helping neutralize any remaining acid and providing buffering in the distal intestine. Potassium can be both absorbed and secreted, with the colon being a significant site of potassium secretion. Secretory diarrhea occurs when chloride secretion exceeds sodium absorption, creating an osmotic gradient that draws water into the lumen. Cholera toxin causes severe secretory diarrhea by locking chloride channels open, producing voluminous watery stools that can rapidly lead to life-threatening dehydration. Understanding the mechanisms of fluid and electrolyte transport has enabled development of oral rehydration therapy that saves millions of lives annually. The colonic microbiota's metabolic activities significantly impact host nutrition and health. Bacteria ferment indigestible carbohydrates, dietary fiber that reach the colon. This fermentation produces short-chain fatty acids, particularly acetate, propionate, and butyrate, along with gases, including hydrogen, carbon dioxide, and methane. The short-chain fatty acids are absorbed by colonocytes and used as energy sources, with butyrate being particularly important for colonocyte health. Butyrate serves as the preferred fuel for colonocytes, providing 70% or more of their energy. It also has anti-inflammatory effects, strengthens the intestinal barrier, and may have protective effects against colorectal cancer. The production of adequate butyrate depends on consuming sufficient fermentable fiber and maintaining a healthy microbiota. This illustrates how diet, microbiota, and intestinal health interconnect. The microbiota also synthesizes certain vitamins, particularly vitamin K and several B vitamins, including biotin, folate, and B12. While bacterial B12 synthesis occurs in the colon beyond the site of B12 absorption, and therefore does not significantly contribute to B12 nutrition, bacterial vitamin K synthesis is meaningful. People with severely disrupted microbiota from prolonged antibiotic use can develop vitamin K deficiency and coagulation problems. The composition of the gut microbiota varies among individuals and populations, influenced by diet, genetics, antibiotic use, mode of birth, and many other factors. Western diets high in processed foods and low in fiber tend to promote different bacterial populations than traditional diets rich in plant fiber. These microbial differences may contribute to variations in obesity risk, inflammatory conditions, and other health outcomes, though the causal relationships remain under investigation. The immune tolerance toward commensal bacteria represents a remarkable achievement. The immune system could easily recognize the trillions of bacteria in the colon as foreign and mount inflammatory responses that would be catastrophic. Instead, multiple mechanisms maintain peaceful coexistence. The mucus layer physically separates bacteria from the epithelium. Pattern recognition receptors distinguish pathogenic from commensal bacterial signals. Regulatory immune cells actively suppress inflammatory responses to commensals. This tolerance develops through ongoing interactions between microbiota and immune system, particularly in early life. The gut-brain axis, that bidirectional communication network between digestive system and central nervous system, involves multiple pathways. The vagus nerve carries signals in both directions. Sensory information from gut to brain about distension, nutrients, and inflammation, and motor commands from brain to gut affecting motility and secretion. Gut hormones like CCK, ghrelin, and peptide YY enter the bloodstream and affect brain regions, controlling appetite, mood, and cognition. The microbiota participates in gut-brain communication by producing neurotransmitters and other signaling molecules. Gut bacteria produce gamma-aminobutyric acid, serotonin, dopamine, and other neurotransmitters, though whether these directly affect the brain or primarily act locally remains debated. Bacterial metabolites can influence the enteric nervous system and send signals to the brain through vagal pathways. Some evidence suggests that gut microbiota composition can influence anxiety, depression, and even cognitive function, though mechanisms remain incompletely understood. Stress demonstrates the brain's influence on the gut. Acute stress triggers the fight or flight response, with sympathetic activation that inhibits digestive functions. Blood flow is redirected from splanchnic organs to skeletal muscles. Motility changes, often slowing in stomach and small intestine while accelerating in colon, sometimes causing stress-induced diarrhea. Chronic stress can alter gut barrier function, increase intestinal permeability, and promote inflammation. The relationship is bidirectional. Gut inflammation and dysfunction can feed back to influence stress responses and mood. Functional gastrointestinal disorders like irritable bowel syndrome demonstrate this gut-brain interaction. Patients experience abdominal pain, altered bowel habits, and other symptoms without identifiable structural or biochemical abnormalities. The symptoms appear to arise from altered gut-brain communication, visceral hypersensitivity, where normal intestinal sensations are perceived as painful, altered motility patterns, and psychological factors like anxiety or depression. Treatment often requires addressing both gut symptoms and psychological components. The gastrointestinal system's integration with circadian rhythms reveals another dimension of regulatory sophistication. Digestive functions show daily rhythms coordinated by the body's master clock in the suprachiasmatic nucleus. Gastric acid secretion peaks in the evening and early night. Colonic motility shows circadian variation, with mass movements more common in morning hours. Hepatic metabolic processes vary throughout the day. The microbiota composition and metabolic activity also show circadian fluctuations. These rhythms coordinate digestive function with anticipated feeding times. Disruption of circadian rhythms, as occurs with shift work or jet lag, can disturb digestive function. Some evidence suggests that eating at inappropriate circadian times, late-night meals when digestive efficiency is naturally lower, may contribute to metabolic dysfunction. The timing of meals relative to circadian phase may matter as much as meal composition for metabolic health. The gastrointestinal system's responses to aging demonstrate both robustness and vulnerability. Many digestive functions remain adequate throughout life in healthy individuals. Others show decline. Taste perception often decreases with age, reducing appetite and potentially affecting nutrition. salivary production may decrease, affecting initial digestion and oral health. Gastric acid secretion tends to decrease, which can impair mineral absorption and increase risk of bacterial overgrowth. Intestinal motility may slow with age, increasing constipation risk. The microbiota composition shifts, generally showing reduced diversity in elderly individuals. Immune function in the gut may decline, potentially increasing infection and inflammation risk. However, many of these changes are not inevitable and can be influenced by diet, physical activity, and overall health status. The remarkable regenerative capacity of the intestinal epithelium deserves emphasis. The epithelium completely renews every 3 to 5 days through continuous proliferation of stem cells in the crypts. These stem cells divide, producing daughter cells that migrate up the villi while differentiating into the specialized cell types, enterocytes, goblet cells, enteroendocrine cells. This rapid turnover allows the epithelium to repair damage quickly and adapt to changing conditions. It also makes the intestinal epithelium vulnerable to chemotherapy and radiation, which target rapidly dividing cells and can severely damage the gut lining. The integration of all these processes, digestion, absorption, secretion, motility, immune function, microbial interactions, neural and hormonal regulation, circadian coordination, creates a system of extraordinary complexity operating seamlessly to extract nutrients from food and maintain the barrier between self and environment. Each component depends on others, each influences others, creating emergent properties that transcend individual parts. As you rest here, your gastrointestinal system continues its ceaseless work. Smooth muscle contracts rhythmically. Enzymes catalyze reactions. Nutrients cross epithelial barriers. Immune cells patrol for threats. Bacteria ferment fiber. Hormones coordinate activities. Neural signals adjust functions. All these processes unfold automatically, reliably, maintaining the flow of energy and materials that sustains your existence. The river of digestion flows on through its ancient course, shaped by millions of years of evolution, adapted to transform the external world into the substance of life itself. This transformation continues moment by moment, meal by meal, as the gastrointestinal system performs its essential service of bridging the gap between environment and organism, between food and self. As we arrive at this final passage through the gastrointestinal system's territories, we find ourselves not at an ending, but at a deeper understanding of beginnings. How life sustains itself. How the external becomes internal. How the simple act of eating connects to the vast web of physiological processes that define human existence. The digestive system we have explored is not merely a biological apparatus, but a living interface between organism and world, a transformer of matter, a guardian of boundaries, a participant in the most fundamental transaction of life. The evolutionary perspective illuminates the digestive system's design with particular clarity. The gut represents one of the oldest organ systems, present in primitive form even in the earliest multicellular animals. The basic plan, a tube running through the body with specialized regions for different functions, appeared hundreds of millions of years ago and has been refined through countless generations. Natural selection has sculpted digestive systems to match dietary niches, producing the short, simple guts of carnivores, the complex, multi-chambered stomachs of ruminants, the elongated ceca of herbivores fermenting plant material. The human digestive system reflects our omnivorous heritage. We lack the specialized fermenting chambers of dedicated herbivores, but retain a functional cecum and colon harboring fiber fermenting bacteria. We produce enzymes for digesting both plant and animal materials. Our teeth include incisors for cutting, canines for tearing, and molars for grinding, suited for varied diets. Our intestinal length falls between the extremes of carnivores and herbivores. This anatomical flexibility mirrors the dietary flexibility that allowed human ancestors to exploit diverse food sources and ultimately colonize nearly every terrestrial environment. The capacity to digest starch, unusual among primates, appears to have increased during human evolution. Multiple copies of the amylase gene provide higher salivary amylase levels than most other primates, suggesting that starchy foods became important in human diet. The ability to digest lactose into adulthood evolved independently in several populations that practice dairy farming, providing a recent example of human digestive adaptation to cultural practices. These evolutionary changes demonstrate how diet and digestion shape and are shaped by each other. The developmental journey from conception to adulthood reveals the digestive system's gradual maturation. The embryonic gut begins as a simple tube that undergoes dramatic folding and specialization. The midgut elongates rapidly and must herniate temporarily out of the abdominal cavity before returning and rotating into its final position. Failure of proper rotation or return can cause malformations requiring surgical correction. The hindgut gives rise to the colon and rectum. The foregut forms esophagus, stomach, and the proximal duodenum, while also budding off the liver, pancreas, and biliary system. At birth, the digestive system must rapidly adapt to enteral nutrition after months of receiving nutrients through the placenta. The newborn's first feeding initiates a cascade of changes. Digestive enzyme production increases. Motility patterns mature. The sterile gut becomes colonized with bacteria, beginning the development of the microbiota. Breast milk provides not only nutrition, but also immune factors, growth factors, and prebiotics that support beneficial bacteria and promote intestinal development. The infant digestive system differs functionally from the adult system. Lactase activity is high, allowing milk sugar digestion. Pancreatic amylase production is low initially, limiting starch digestion. The stomach produces less acid. Intestinal permeability is relatively high, allowing absorption of intact antibodies from colostrum that provide passive immunity. These adaptations suit the infant's milk-based diet and gradually shift as solid foods are introduced. The introduction of complementary foods represents a crucial transition. The digestive system must adapt to new nutrients, textures, and antigens. The microbiota diversifies as new substrates become available. Immune tolerance mechanisms engage with food proteins to prevent allergies. The timing and manner of introducing solid foods may influence long-term digestive and immune health, though optimal practices remain debated. Throughout childhood, digestive capacity increases with body size. Enzyme production matures. The microbiota becomes more stable and adult-like. Eating patterns and food preferences develop through learning and cultural transmission. The digestive system generally reaches functional maturity by adolescence, though the microbiota may continue evolving into early adulthood. The pathophysiology of digestive disorders illustrates what occurs when the system's intricate balance is disrupted. Peptic ulcer disease, once attributed primarily to stress and spicy foods, is now understood to result mainly from Helicobacter pylori infection, or from non-steroidal anti-inflammatory drugs damaging the protective mucus layer. The bacterium H. pylori produces urease that generates ammonia, neutralizing stomach acid locally and allowing the bacterium to survive in the acidic environment. The infection triggers inflammation and can erode the protective barriers, allowing acid and pepsin to damage the underlying tissue. Inflammatory bowel diseases. Crohn's disease and ulcerative colitis involve inappropriate immune responses to the gut microbiota in genetically susceptible individuals. The immune system attacks the intestinal lining, causing chronic inflammation, ulceration, and various complications. Ulcerative colitis affects only the colon, causing continuous inflammation of the mucosa. Crohn's disease can affect any part of the digestive tract and involves transmural inflammation extending through all layers of the intestinal wall. Both conditions demonstrate how dysregulated immune microbial interactions can cause severe intestinal pathology. Celiac disease represents an immune reaction to gluten proteins in wheat, barley, and rye. In susceptible individuals with specific genetic variants, gluten peptides trigger an immune response that damages the small intestinal villi, causing villus atrophy. The flattened mucosa has greatly reduced absorptive surface area, leading to malabsorption of nutrients. Symptoms can include diarrhea, abdominal pain, and deficiencies of various nutrients. The only effective treatment is strict avoidance of gluten, allowing the villi to regenerate. Lactose intolerance, affecting the majority of the world's adult population, results from declining lactase production after weaning. Undigested lactose reaches the colon, where bacteria ferment it, producing gas and organic acids that draw water into the lumen, causing bloating, cramping, and diarrhea. Populations with a long history of dairy consumption tend to maintain lactase production into adulthood through genetic adaptation. This lactase persistence represents one of the clearest examples of recent human evolution driven by cultural practices. Gastroparesis, delayed gastric emptying, can result from diabetes damaging the nerves controlling stomach muscles, from certain medications, or from unknown causes. Food remains in the stomach far longer than normal, causing nausea, vomiting, bloating, and unpredictable blood glucose control in diabetics. The condition illustrates the importance of proper motility timing for digestive function and comfort. Small intestinal bacterial overgrowth occurs when bacteria proliferate excessively in the small intestine, where bacterial populations are normally relatively low. The bacteria compete for nutrients, produce gas causing bloating and discomfort, and can damage the intestinal lining. Conditions that slow small intestinal transit, create blind loops, or impair the migrating motor complex can predispose to overgrowth. Treatment typically involves antibiotics to reduce bacterial load. Colorectal cancer represents one of the most common malignancies in developed countries. Most cases arise from adenomatous polyps that undergo malignant transformation over many years. Screening programs that detect and remove polyps can prevent cancer development. Risk factors include age, family history, inflammatory bowel disease, and dietary patterns high in processed meat and low in fiber. The adenoma to carcinoma sequence has been well characterized, providing insights into cancer development and prevention. The liver's vulnerability to various insults reflects its central role in metabolism and detoxification. Viral hepatitis, caused by several different viruses, can cause acute or chronic liver inflammation. Hepatitis B and C can lead to chronic infection, cirrhosis, and liver cancer. Alcohol-related liver disease progresses from fatty liver to alcoholic hepatitis to cirrhosis through mechanisms involving direct toxic effects of alcohol and its metabolites, inflammation, and fibrosis. Non-alcoholic fatty liver disease, increasingly common with rising obesity rates, can similarly progress to inflammation and fibrosis without alcohol consumption. Cirrhosis, the end stage of chronic liver disease, involves widespread fibrosis and nodular regeneration that disrupts normal liver architecture. Portal hypertension develops as scarring impedes blood flow through the liver, causing blood to back up in the portal system. This can lead to varices, dilated veins in the esophagus and stomach that can rupture and bleed catastrophically. Ascites, fluid accumulation in the abdomen, develops from the combination of portal hypertension, decreased albumin production, and altered renal function. Hepatic encephalopathy occurs when the failing liver cannot adequately remove toxins like ammonia, which accumulate and affect brain function. Gallstone disease demonstrates how alterations in bile composition can cause pathology. When cholesterol concentration exceeds the solubilizing capacity of bile salts and lecithin, cholesterol can precipitate and form stones. Pigment stones form from bilirubin. Most gallstones remain asymptomatic, but if stones obstruct the cystic or common bile duct, they cause pain and potentially serious complications, including cholecystitis, pancreatitis, or ascending cholangitis. Acute pancreatitis, often caused by gallstones or alcohol, involves premature activation of pancreatic enzymes within the pancreas, leading to autodigestion of pancreatic tissue and severe inflammation. The condition can range from mild and self-limiting to severe and life-threatening with multi-organ failure. Chronic pancreatitis, typically from prolonged alcohol use, causes progressive destruction of pancreatic tissue, leading to loss of both exocrine function with maldigestion and endocrine function with diabetes. Nutritional science and the digestive system are inextricably linked as the gut mediates the relationship between diet and health. Macronutrients, carbohydrates, proteins, and fats provide energy and building blocks. Micronutrients, vitamins, and minerals enable countless biochemical reactions. Fiber, though indigestible by human enzymes, provides substrate for microbial fermentation and influences gut health. Water supports all bodily functions and is absorbed primarily in the intestines. The concept of a balanced diet reflects the need to provide adequate amounts of all essential nutrients while avoiding excess of any particular component. Different macronutrient ratios suit different individuals and circumstances. Some people thrive on higher carbohydrate intake, others on higher fat intake. The digestive system can adapt to these variations through changes in enzyme production, transporter expression, and other adjustments. The glycemic index and glycemic load concepts recognize that different carbohydrate-containing foods affect blood glucose differently. Rapidly digestible starches and simple sugars cause quick glucose spikes. Slowly digestible starches and fiber-rich foods produce more gradual glucose rises. The digestive and absorptive processes that determine these differences have important metabolic implications, particularly for insulin secretion and diabetes risk. Protein quality depends on amino acid composition. Complete proteins contain all essential amino acids in adequate proportions. Most animal proteins are complete, while many plant proteins are incomplete, lacking adequate amounts of one or more essential amino acids. However, combining different plant proteins can provide complete nutrition, as cultures relying on plant-based diets have long understood through combinations like rice and beans. Fat quality involves the types of fatty acids consumed. Saturated fats, monounsaturated fats, polyunsaturated fats, and trans fats have different health effects. Essential fatty acids, certain omega-3 and omega-6 polyunsaturated fats, cannot be synthesized and must come from diet. The digestive system absorbs all these fats similarly, but their subsequent metabolic effects differ substantially. The role of fiber in digestive health has gained increasing recognition. Soluble fiber dissolves in water, forming gels that slow gastric emptying and glucose absorption, bind cholesterol, and provide substrate for beneficial bacteria. Insoluble fiber adds bulk to stool, speeds intestinal transit, and promotes regularity. Most plant foods contain both types. Adequate fiber intake supports healthy gut motility, beneficial microbiota, and may reduce risks of various diseases. The gut microbiota's influence on nutrition extends beyond vitamin synthesis and fiber fermentation. Bacteria can metabolize plant polyphenols and other compounds, releasing bioactive metabolites that humans absorb. Different bacterial species have different metabolic capabilities, so microbiota composition influences how much nutrition we extract from food. This helps explain why individuals eating identical diets may experience different metabolic outcomes. Prebiotics, dietary components that selectively promote beneficial bacteria, and probiotics, live bacteria consumed to confer health benefits, represent attempts to modulate the microbiota for health. Prebiotics include certain fibers like inulin and fructooligosaccharides. Probiotics include specific strains of bacteria, commonly Lactobacillus and Bifidobacterium species. Evidence for health benefits varies by specific prebiotic or probiotic and health outcome, with some applications showing clear benefits and others remaining uncertain. The concept of personalized nutrition recognizes that optimal diets may vary among individuals based on genetics, microbiota composition, metabolic status, and other factors. Some people metabolize caffeine rapidly, others slowly. Some maintain normal blood pressure despite high salt intake. Others are salt sensitive. Some people's glucose rises sharply after eating bread, others does not. Understanding these individual differences may allow tailored dietary recommendations, though practical implementation remains challenging. The social and psychological dimensions of eating extend beyond the digestive system's physiology, but profoundly affect it. Meals serve social functions, bringing people together, marking celebrations, expressing culture. Food preferences and aversions develop through experience and learning. Eating disorders, anorexia nervosa, bulimia nervosa, binge eating disorder, involve disturbed relationships with food and eating that can severely impact digestive and overall health. The modern food environment, with its abundance of highly palatable, calorie-dense, processed foods, creates challenges for which the digestive system has not evolved. The system evolved in environments of scarcity and variety, where energy-dense foods were rare and valuable. Today's easy access to concentrated calories contributes to obesity and metabolic disease epidemics. Understanding digestive physiology helps clarify why maintaining healthy eating patterns requires conscious effort in modern environments. Fasting and time-restricted eating represent practices that alter the temporal pattern of digestive activity. Prolonged fasting shifts the body from fed to fasted metabolism, with the digestive system becoming quiescent, the migrating motor complex activating to clear the intestines, and metabolic reliance shifting toward stored energy. Time-restricted eating, limiting food intake to specific hours, may synchronize eating with circadian rhythms and allow extended daily fasting periods. Research continues to explore potential health benefits of these practices. The future of gastroenterology and digestive health research promises new insights and interventions. Microbiome science continues revealing connections between gut bacteria and health. Techniques for analyzing microbiota composition and function are advancing rapidly. Fecal microbiota transplantation, transferring gut bacteria from healthy donors to recipients, has proven remarkably effective for recurrent Clostridioides difficile infection and is being explored for other conditions. Advances in understanding the gut-brain axis may lead to new treatments for both digestive and neuropsychiatric conditions. The recognition that many neurotransmitters and signaling molecules operate in both gut and brain suggests potential for novel therapeutic approaches. The vagus nerve has become a target for neuromodulation in various conditions. Regenerative medicine approaches aim to repair or replace damaged digestive tissues. Stem cell therapies might regenerate pancreatic islet cells in diabetes or repair damaged intestinal epithelium in inflammatory conditions. Tissue engineering could potentially create replacement tissues or even whole organs. While such approaches remain largely experimental, they represent promising directions. Precision medicine, tailoring treatments to individual characteristics, is advancing in gastroenterology. Genetic testing can identify individuals at high risk for certain cancers or those with specific genetic disorders affecting digestion. Pharmacogenetics helps predict individual responses to medications. Microbiota profiling might guide personalized dietary recommendations or targeted probiotic interventions. As we complete this comprehensive exploration, we return to where we began, to the present moment, to your body, to the quiet processes unfolding within. The gastrointestinal system we have examined in such detail is not an abstraction, but a living reality within you right now. Smooth muscle fibers are contracting and relaxing in coordinated waves. Enzymes are catalyzing reactions, breaking bonds, transforming molecules. Nutrients are crossing epithelial barriers, entering your bloodstream, becoming part of you. Perhaps you sense something of this activity, a gentle gurgling, a subtle fullness or emptiness, a warmth in your abdomen. These faint sensations represent the sensory surface of vast coordinated processes. Beneath conscious awareness, billions of cells are performing their specialized functions. Trillions of bacteria are metabolizing fiber, producing vitamins, communicating with immune cells. Neural networks are integrating information and adjusting activities. Hormones are circulating, signaling between organs. The river of digestion flows through its eternal course, from mouth to exit, transforming food into you. Every molecule of glucose energizing your brain cells, every amino acid building your proteins, every fatty acid forming your cell membranes, every vitamin enabling your enzymes. All these entered through this system. You are quite literally made from what you have eaten, transformed through digestion into living tissue. This transformation represents one of life's fundamental miracles, the ability to incorporate the external world into the self, to find in the environment the materials for building and maintaining the body. Plants capture sunlight and carbon dioxide, building organic molecules. Animals eat plants or other animals, breaking down those molecules and rebuilding them into animal tissue. Humans continue this great chain, extracting from food the energy and materials that sustain us. The gastrointestinal system's elegance lies in how seamlessly it performs this transformation. The mechanical processing, chemical digestion, selective absorption, immune surveillance, microbial partnerships, neural and hormonal regulation, all these elements work together so smoothly that we can eat a meal and trust that the necessary processes will unfold. We need not consciously direct enzyme secretion or control intestinal absorption. The system's intelligence operates autonomously, reliably, ceaselessly. This autonomy mirrors the autonomy of breathing, of heartbeat, of all the involuntary processes that sustain life. Yet the digestive system also allows voluntary control through food choices, eating patterns, and feeding behaviors. We choose what to eat, when to eat, how much to eat, exercising agency over the inputs while the processing remains automatic. This blend of voluntary and involuntary, of conscious choice and unconscious execution, characterizes the digestive experience. The sensory pleasures of eating, the tastes, aromas, textures that make eating enjoyable, serve biological purposes in guiding food selection, but they also make eating one of life's great pleasures. The gastrointestinal system thus connects not only to nutrition and metabolism, but also to pleasure, satisfaction, and the cultural and social dimensions of food. It is a system thoroughly integrated into the fullness of human experience. The system's robustness across decades of use demonstrates remarkable durability. The continuous renewal of the intestinal lining allows rapid repair of damage. The redundancy and reserve capacity mean that substantial damage can occur before function becomes inadequate. Many people reach advanced age with digestive systems functioning adequately despite a lifetime of varied diets, occasional insults, and the gradual changes of aging. Yet the system is also vulnerable, subject to infection, inflammation, malignancy, and numerous disorders that can impair function and quality of life. This vulnerability reminds us that digestive health requires care, thoughtful food choices, adequate hydration, attention to symptoms, appropriate medical care when needed. The ancient wisdom of various cultures regarding food, eating practices, and digestive health often aligns with modern scientific understanding, suggesting deep empirical knowledge accumulated over generations. In our modern era, we possess unprecedented scientific understanding of digestive physiology, yet we also face unprecedented challenges: processed foods, sedentary lifestyles, antibiotic overuse, environmental toxins. The digestive system, shaped by millions of years of evolution for an environment very different from today's, must adapt to these novel conditions. Supporting digestive health in modern contexts requires both understanding the system's evolved design and recognizing contemporary challenges. As this exploration concludes, as we complete this circuit from mouth to exit and back to awareness of the whole, recognize that the knowledge we have traversed is not merely academic, but deeply personal. This is your digestive system, your gut, your microbiota, your liver, your pancreas. These structures and processes enable your existence. Understanding them more deeply creates opportunity for appreciating their function, for making choices that support their health, for recognizing symptoms that might warrant attention. The gastrointestinal system continues its work, whether you think about it or not, whether you understand it or not, whether you appreciate it or not. It has functioned faithfully since you first suckled as an infant, will continue through all your days, transforms your food into your substance, maintains the boundary between self and world, participates in immunity and metabolism and cognition. It is essential, complex, elegant, resilient, and worthy of appreciation. Rest in the knowledge that you are sustained by systems of extraordinary sophistication and reliability. The river of digestion flows through you, connecting you to the earth that grows your food, to the sun that energizes all life, to the long history of evolution that shaped digestive physiology, to all living things that likewise extract nourishment from their environments. You are not separate from nature, but deeply embedded in it, sustained by it, part of it. The food you ate yesterday is becoming you today. The food you will eat tomorrow will become you next week. This continuous transformation, this ongoing exchange of matter between organism and environment, defines biological existence. The gastrointestinal system mediates this exchange with precision and grace, allowing you to live, to grow, to maintain yourself, to continue your journey through time. Even now, as these words conclude, the digestive processes continue. Nutrients are being absorbed. Waste is being prepared for elimination. The microbiota ferments fiber. Hormones coordinate distant organs. The enteric nervous system monitors and adjusts. Epithelial cells renew themselves. Life sustains itself through these quiet, faithful processes that ask nothing and give everything. The river flows on, ancient and ever new, carrying the materials of existence from world to body, from food to self, from one meal to the next, one day to the next, one life to the next. This is the way life works, the way it has always worked, the way it will continue working. The gastrointestinal system, that long and winding passage through the body's core, makes it all possible.